Good morning to you all learners. I am Professor S. Kripanidhi, heading the Department of Biotechnology at Vignan's Foundation for Science, Technology and Research at Deemed University, located in a rural vicinity, Vadlamudi, Guntur district, Andhra Pradesh, India, are giving a series of talks on and immunoinformatics. Covered 2 units and now antigen antibody reactions, precipitation, and agglutination. The third unit predominantly deals with the experimental protocols and experimental procedures and the results outcome of the immunological laboratory sessions. Okay, so let us see now. See, as we have gone through earlier, antigen is the one that elicites immune response. Okay, in an individual, antigen elicits immune response. As a result, there is a T cell response, there is a B cell response, there is immunoglobulins that are released, there is humoral activity, several things. But to visualize this immune response, we have to do some of the experiments in vitro. So such type of the experiments we are planning, and those details will be shown now. Okay. So what is the experiment? That means why antigen has to react with the antibody? Why not antigen react with some other substance? Because it is highly specific. Anti-mono, that's what we have seen hybridoma technology, where a monoclonal antibodies production. And we also have seen the market potential, pharma market potential of this monoclonal antibody production. So these are the monoclonal antibodies which are highly specific to the epitope of the antigen, that is highly reactive. There are binding forces involved. Binding forces are involved between the antigen and the antibody. That's what those details we see in vitro also now. Okay, let us see.
Hi, in this video, we would quickly describe the concept of affinity and avidity of antigen-antibody interaction. So these two terms determines the interaction between antigen and antibody. Not only interaction, the strength of interaction and the degree which and also the kinetics of the interaction between antigen and antibody. So antigen and antibody interacts in multiple non-covalent way, and all these interactions are reversible. So most prevalent interactions are hydrogen bond, ionic interactions, then hydrophobic interactions, and van der Waals interactions. All these are relatively weak interactions and they are reversible. So affinity is a measure of the strength of interaction between the epitope, that means the specific antigenic determinant, and the antibody's antigen binding site. So between these two, the binding strength is known as affinity. Now, affinity could be defined or could be described in the simple thermodynamic principle which governs the biomolecular reversible interactions. So, assuming there is antigen and antibody is making a complex, and these complex could be broken down as well, and that is why this interaction is reversible, and this is denoted by simple equation like mentioned here. So the equilibrium association constant would be simply defined like this, and the dissociation on the other hand would be determined like this, and a ratio of association versus dissociation would give the binding affinity of an antigen antibody reaction. Now this antigen and antigen antibody interaction is happening at a molecular level. So we were talking about a microscopic level, which would, and the measure of this microscopic interaction is affinity. Now let's just talk about at a macroscopic level. That means antibody could have multiple different antigen binding sites. I mean that is determined by its valency. For example, IgA has 4 antigen binding site, where IgM has 10. So the overall interaction between all of these potential antigen binding site is collectively known as avidity. So we can simply understand avidity as affinity at a macroscopic level. Now, avidity is dependent upon three major parameters. Definitely, affinity of the antibody towards its epitope. So, greater the affinity, greater would be avidity as well. Then, valency of the antibody. So, greater the valency, greater would be avidity. For example, IgM has greater avidity than IgT. And last but not the least is the molecular arrangement of the epitope and the antibody antigen binding site inside the antibody. So this three-dimensional arrangement sometimes.
So just now we are seeing the animated video showing the antigen antibody binding, affinity, avidity variation. IgM is having more avidity because of three factors. One is this valency, the other one is the affinity, the third one is the structural complexity of the IgM. So those details are shown. So that means an antigen and antibody interaction is through affinity and avidity means. Okay, now those details we see now. So the objectives of this particular topic 1 unit 3 antigen antibody interaction is as follows. Okay, so to elucidate the characteristics of antigen antibody interaction, to familiarize with the forces that made to bind antigen antibody interactions, and to notice the experimental results on the antigen antibody precipitation and agglutination. Precipitation and agglutination. Okay. So whenever there is a patient with jaundice, if they go to the doctor, doctor will advise to have the clinical examination on antibody titer. Antibody titer. So these are all the various procedures that we are going to explain in this particular topic. Okay, so this is the interaction and the forces that are there which make them bound, antigen and antibody, precipitation and agglutination aspects we are seeing now. Okay, now the next one is the after having gone through around 17 to 18 slides in this particular topic, the learners are going to have the following outcomes. The knowledge on antigen antibody interaction facilitates the learners to plan experiments in the minor research projects for the diagnosis of chosen antigen. We may be interested in a particular antigen and how this antigen elicits immune response, how to harvest antibodies, how to do after-lonely double immunodiffusion immunoelectrophoresis, and how to work out the antibody titers. All those experiments one can plan once you go through the slides. Okay, so it will be useful for planning your minor research projects in the regular BTech curriculum. So now, let us see the characteristics of the antigens. Antigens are having immunogenicity. That is, they elicit immune response. The property of substance to induce a detectable immune response. Okay, so of course we have seen immunogen and antigen as synonymous. Let us not go into that confusion. Now, so it induces a detectable immune response. Okay, so that's what detectable immune response means. The dose that we give, immunogen dose that we give should be proper enough. Then antigenic specificity. Property of antigen molecule to react with specific antibodies. Okay, so that is what this immunology is now in the limelight. Because it reacts with the specific antibodies, the ELISA have come. ELISA technique, that is enzyme liquid immunosorbent ELISA technique. YALO, a Nobel laureate has identified this radioimmunoassay. Okay, the number of antigenic determinants usually varies with the size and chemical complexity of the macromolecule. So let us say, for example, egg oval women, molecular weight 42,000, it has 5 antigenic determinants. And thyroglobulin has got a molecular weight of 70,000, it is having 40 as many as 40 the ADS, the antigenic determinant sites. So like this, if we take up the hemocyanin, which is an immunogen, it will be having more than 100, what is that called, ADS or epitopes. So the molecular size determines the number of what is that called antigenic determinant sites. Okay. So let us see some more characteristics of the antigen. See now, this is an antibody. This region of the antibody is called FAB, that is fragment antigen binding. Of course, let us not go to the other details which we have covered in the unit 2. So this is the fragment antigen binding. We say label this as FAB. Okay, so let us say this is the antigen. Here the antigen is shown just as spherical, but it is having a lot of spikes around. That is called ADS, antigenic determinant sites. Now let us see, once these two, this is soluble, both got formed antigen-antibody complex, wherein the antigen binds only at Fab region of the immunoglobulin. Fab region of the immunoglobulin, that is fragment antigen binding region only. This is the region where there is complementarity determining factors, and there is hypervariable regions, and all three hypervariable regions are there. Okay, so this antibody doesn't fit into this group, doesn't fit into the FC. Here, this is said to be the variable region. All these 4 points are said to be the variable region. This is what antigen-antibody complex. This complex we are studying in vitro now. So let us see. So if you are having an antigen, let us say this is an antigen. Antigen is having one site called antigen binding site A. This is antigen binding site B. This is antigen binding site C. If there are 3 antigen binding sites on the antigen, the immunoglobulin that is made for antigen binding site A is antibody A. The immunoglobulin that is made for antigen binding B is called antibody B. The immunoglobulin that is made for antigen binding site C is called antibody C. So A, B, C antibodies are quite different in their properties. What is their property? The property of antibody A is binding to antigen binding site of A. It doesn't bind anyway. So the property of antibody C is it binds to antigen binding site C only. Well, that is what it varies. It is made. That's what called immune response. Okay, this region is said to be the epitopes. Antigenic determinants are called epitopes. So they are all given in different shapes. Accordingly, antibodies are made. So let us see the immunological test that we are seeing. A harmful agent can be detected, such as an antigen, a virus. Let us say a bee, an insect bee bites us. It releases some sort of what is it called toxins, viruses, bacteria. So they are hard to culture, and as a result, we can take the advantage of specific nature of antibodies. We take the antibodies and evaluate. So we raise in amount of antibody as an indicator. An increase in specific antibody is an indicator of exposure. So that's what, see please see here, an increase in the specific antibody is an indicator of exposure. That's what now in COVID-19 patients, they test after 14 days, 15 days, the antibody in the serum. If invariably doctor finds there is an increase in the amount of the IgG, so that indicates he has been exposed to COVID-19. That confirms the diagnosis. Antibody binds, precipitates, and agglutinates in the in vitro setup. So when sufficient antigen-antibody molecules interact, they precipitate out. They precipitate out of a solution. Okay. So too few antigen molecules, little precipitate. Too many antigen-antibody cross links not made. Okay. So example, immunodiffusion. So that is one of the simplest, earliest experiment that has been designed here. Antibody-antigen react in agar medium, in agar solution that is made, agar gel, to make a precipitin bands or rings. Okay. So immunoelectrophoresis is also an advanced version of this immunodiffusion with electrical flow of proteins. Complex mixture of antigens can be separated, then reacted with antibody. Okay, now let us see a pictorial representation of this antigen-antibody interaction. So now in a tube, if you put the soluble antigen and soluble antibody, both at the intersection point, they join and form a precipitation zone. Okay, so in this precipitation zone, see the Fab region, this antigen is getting bound. Okay, so that is soluble. Here the precipitation comes in this region, and this protein is soluble. Here antibodies are also proteins, they are soluble. So the next one is C. So now this is an interesting observation in the experiments it has been shown. See, this is antigen excess, antigen less and antibody excess zone. Antibody excess zone. This is antibody, this is antibody. So here excess antibody is not being engaged by this antigen. Here more of antigen zone, excess antigen zone. Okay, so some of these antigens are unable to bind to the antibodies. Here, this is an equivalent zone where the antibody, antigen both are bound. So that's what here access in equivalent zone will be getting good amount of the precipitate. Clear? So this again, once the immune complexes lattice is formed, so that's what the binding shown here. So this is what the precipitation reaction. Antibody, antigen, a lattice is formed. That's what the precipitation reaction. And again, this is agglutination reaction. Precipitation goes with the antigen antibody separately if we take up and work out with the in vitro studies. But with regard to the agglutination, this is bacteria. We are bringing the bacteria against the bacteria. Already antibodies are available with us, and if those antibodies are made to interact with the bacteria, bacterial agglutination you can see. Bacterial agglutination one can see through the microscope. This is what the agglutination, the earlier one is the precipitation. Let us go to the details. Precipitation test. Antigen concentration. Antigen concentration, if it is excess, okay, so there is no much precipitation. If there is less, there is no much precipitation. Maximum precipitation occurs where there is equivalence zone. Okay, so usually in the laboratory, this is all the theory behind, and the theory has come from the experiments. This is what the precipitation reaction. This is what called agarose gel. Agar-agarose gel, wherein we are having the peripheral wells and the central well. This is usually set with Ochterlony double immunodiffusion. In the center, we keep what is called the antibody. In the peripheral wells, we are going to keep the antigens so that they diffuse. In the process of the diffusion, we are going to have what is this called crescentic. This is a crescentic arc. Okay, half moon shaped type of arcs come. Half moon shaped type of arcs will come. So these are all the various procedures. In this also we will work out whether the antigen is closely related or far related. If far related, there is no spur formation. If it is antigen is closely related, spur formation comes. Such type of things also one can do in the minor research project that is designed in our experiments. Okay. Now let us see in this case a little more detail. Sensitivity and specificity. The ability to test, to detect very small amounts of antigen, that is analytical sensitivity. Chemical sensitivity is ability to test to give positive results if patient has the disease. Okay. Analytical specificity means ability to test to detect substances without interference from cross-reacting substances. So that's what analytical specificity. Clinical specificity means the ability to test to give negative result if patient does not have the disease. That's what the clinical specificity. Even if a few of the corona people, they are corona infected, we are getting the diagnosis as negative. A few of them who are positive, at times you get the negative. That's what the clinical specificity, analytical specificity is very much essential. Okay. Now in the video also we have seen affinity and avidity. Okay, let us go into the details of affinity avidity now. Affinity refers to the strength of the binding between a single antigenic determinant and an individual antibody. That is, every antibody is made against one antigenic determinant. That's called epitope. That is the strength. That's what we study. Affinity is the equilibrium constant that describes antigen-antibody reaction. Antibody affinity is the strength of the reaction between a single antigenic determinant and a single combining site on the antibody. Combining site on the antibody is called the paratope. Okay, combining site on the antigenic determinant is called epitope. It is the sum of the attractive and repulsive forces operating between the antigenic determinant and the combining site that makes the affinity. Okay, now ultimately let us know here these two are binding means because of the amino acids. Paratope of the immunoglobulin, epitope of the antigen, both of them are having chargeable amino acids. those chargeable amino acids will bind. That's what here ultimately the affinity. Okay, there is no question of other things. It's only amino acid, one amino acid, another amino acid interaction. Avidity. Avidity is a measure of the overall strength of binding of an antigen with many antigenic determinants and multivalent antibodies. Multivalent antibodies. A multivalent antibody is IgM. Avidity is influenced by both the valence of the antibody and the valence of the antigen. Okay, so if the valence of the antigen is more, that is hemocyanin is having more, then it is avidity that makes it to get bound. Avidity is more than the sum of the individual affinities. Okay, so let us see this diagram. So now if we take up FAB, that is fragment antigen binding, IgG, one more IgG and IgG. So fragment antigen binding, effective valence is only one, here only it binds. So IgG, it's only shown here, one FAB is bound, another FAB is not bound to the antigen. So if you take up this IgG, both the FABs are bound with that of the antigen. So here valence is 2. If you take up the IgM, here it is pentamerous. Okay, each of these FAB is bound to the, but here 1, 2, 3, 4, 5. 5 are bound, so that 5 is effective valence. Okay, so that's what the affinity avidity means. So affinity refers to the strength of the binding between a single antigenic determinant and an individual antibody combining site. Okay, affinity is the equilibrium constant that describes the antigen-antibody reaction. Antibody affinity is the strength of the reaction between a single antigenic determinant and a single combining site on the antibody. It is the sum of the attractive and repulsive forces operating between antigenic determinant and the combining site. What are the attractive, what are the repulsive forces? We see in the next slides. So now let us see the titer. This is what whenever anybody is suffering from the hepatitis B virus, doctor advises you to do the titer. How to do the titer? So that is directly we take the serum, and the serum is tested in a clinical test with the antigen. Okay, so the reaction goes in such a way that, let us see, the serum is getting diluted, one volume of the serum, one volume of the diluted. Okay, this, let us say this is the serum, this is the diluted in all. So this is being shifted like this, shifted like this. So after having gone like this, we'll take at each step we take and do the antigen and antibody interaction test at every, what is that called, tube. So in this particular case, titer 128. Titer 128, 1 by 128. Okay, so here we are getting antigen antibody complex formation interaction. So this is what whenever the clinicians reports to the doctor, titer here is 128. Accordingly, the doctor takes the decision whether it is less antibody in the serum or more antibody in the serum. Doctor will advise. So like this, the titer is being calculated. So summary of this particular topic: electrophoretic. That is, these are all the various forces involved. It's not some various forces involved. Electrostatic force, hydrogen bonding force, hydrophobic force, and van der Waals forces. These are the forces which make the antigen-antibody interaction. Okay, so this is what the rocket immunoelectrophore is, through which also we can evaluate the antigen-antibody interaction. This is immunoelectrophores, wherein various sorts of the crescentic arcs that appear. This also gives the number of antibodies that is produced against antigen. Okay, the forces binding antigen to antibody, as it is shown earlier: electrostatic force, hydrogen binding force, hydrophobic force, and van der Waals force. Let us see the summary of this particular topic. The characteristics of antigen-antibody interaction are elucidated. The forces binding antigen antibody are elaborated. The behavior of antigen-antibody in an in vitro condition is shown. Okay, the study questions: what are Arterloni double immunodiffusion and immunoelectrophores? Explain the characteristics of antigen-antibody interaction. Describe the forces that bind antigen-antibody. Elaborate on the precipitation reactions. Okay, so these are the 4 questions just like that given for you to work at home. Okay, thank you all for your patient listening. We'll see for the next topic sooner. Thank you, one and all.